Welcome back to Fun Facts Online. Today, we are diving deep into a story that sounds like science fiction, or perhaps a kitchen experiment gone wrong, but is actually one of the most significant breakthroughs in optical physics and biology in recent years. We are talking about biological invisibility. For centuries, the idea of seeing through the human body has been a constant obsession. From the myths of magical rings to the terrifying bandages of the invisible man, we have always wanted to see what is hidden. In medicine, this is not just a fantasy, it is a necessity. The human body is opaque. Our skin is an evolutionary marvel, designed to block sunlight and pathogens, but that also means it blocks our eyes. To see a tumor, a blocked vein, or a firing neuron, we usually have to cut the body open or rely on digital approximations like MRIs and CT scans. But what if you could just look? What if you could wipe a lotion on your arm and see the bones and blood vessels beneath, as clearly as looking through a window? In September 2024, a team from Stanford University and the University of Texas at Dallas achieved exactly this. They rendered the skin of a living mouse temporarily transparent. They didn't use dangerous radiation. They didn't use million-dollar equipment. They used a common food dye called Tartrazine, the exact same molecule that gives Doritos and Mountain Dew their bright orange color. This isn't a parlor trick. It is a fundamental hack of the physics of light, relying on a deep principle known as the Kramers-Kronig relations. Over the next 20 minutes, we are going to break down exactly how this works, why your body is opaque in the first place, and the heated scientific debate that this orange dye has sparked. To understand how they made a mouse invisible, we first have to understand why we are visible. Why is skin opaque, while a jellyfish is clear? Why is milk white, even though it is mostly water? The answer is a phenomenon called scattering. Imagine light as a car driving down a road. If the road is clear, the car goes straight. But if you are driving through a dense fog, your headlights hit water droplets and bounce off in every direction. You see a wall of white, not the road ahead. Biological tissue is essentially a dense fog. Inside your body, you are a slurry of different materials. You have water inside your cells, but those cells are wrapped in fatty membranes made of lipids, and they are packed with structures made of proteins. Light travels at different speeds through these different materials. We measure this speed limit using something called the refractive index. In a vacuum, light is at its fastest. In water, it slows down, giving it a refractive index of about 1.33. But fats and proteins are denser. They slow light down even more, having a refractive index of around 1.50. This mismatch is the problem. Every time a photon of light moves from the water in your cell to a protein structure, it changes speed and bends. This is refraction. Because your cells are packed with millions of these tiny boundaries between water and protein, the light bends millions of times in a fraction of a millimeter. It bounces left, right, backward, and forward. This chaotic pinball game is what we call scattering. That is why you can't see your veins through your skin. The light information is scrambled before it can get back to your eye. For over 100 years, scientists have known the solution to this. If you can match the refractive indexes, the scattering stops. If the water and the protein have the same index, the light doesn't bend. It travels straight through, and the object becomes transparent. In the early 20th century, anatomists figured out they could dehydrate dead tissue, removing all the water and replacing it with oils that matched the refractive index of the proteins. This works perfectly. You can turn a dead mouse brain into a glass block. But the catch is obvious. The animal must be dead. You cannot pump oil into a living creature without killing it. Life requires water. So, the holy grail of bioimaging has been to achieve this transparency in a living animal. This brings us to the breakthrough by the Stanford team, led by researchers Guosong Hong, Mark Brongersma, and Zihao Wu. They realized they couldn't remove the water from a living mouse. Instead, they had to change the physics of the water while it was still inside the cells. They needed to raise the refractive index of the water from 1.33 up to 1.50 to match the proteins. Usually, to raise the index of water, you dissolve sugar or salt into it. But to get it that high, you would turn the water into a thick syrup, which would dehydrate and kill the cells immediately. So they turned to the Kramers-Kronig relations. This is a principle in physics that states that a material's ability to absorb light and its ability to bend light are mathematically linked. They are two sides of the same coin. If a material absorbs light very strongly at a specific color, it will drastically change how it bends light at other nearby colors. Think of a calm pond representing the refractive index. If you drop a massive boulder into the water, which represents strong absorption, it creates a splash, but it also sends ripples out across the rest of the pond. The researchers needed a molecular boulder. They found that boulder in tartrazine, also known as FD and C yellow number 5. This dye is incredibly efficient at absorbing blue and ultraviolet light. That is why it looks orange to us. It eats up the blue spectrum. Because it absorbs blue light so violently, the math of Kramers-Kronig dictates that it must raise the refractive index for the longer wavelengths of light, specifically red and yellow light. This was the light bulb moment. By dissolving tartrazine in water, they created a solution that absorbed blue light but raised the refractive index of the water to match the proteins, specifically for red light. When this dye diffused into the skin of the mouse, the water and the cells suddenly had the same optical properties as the fats and proteins. To the naked eye, the mouse looked bright orange. But under red light, the mismatch vanished. The scattering stopped. The skin became a window. Let's walk through the experiment itself. It was elegant in its simplicity. The researchers prepared a topical gel containing the tartrazine and rubbed it onto the shaved skin of sedated mice. At first, the skin was just skin, pale and opaque. As the dye soaked in, the mouse turned that signature Cheeto dust orange. Then, the magic happened. Under the microscope, the fog cleared. When they applied the gel to the scalp, the skull became translucent. They could see the cerebral blood vessels zigzagging across the surface of the brain, mapping blood flow in real time. When they applied it to the abdomen, they could see the liver, the small intestine, and the bladder. They could even watch peristalsis, the rhythmic muscle contractions of the gut moving food along. This is a dynamic, living process that is impossible to see in a dead, cleared specimen. On the leg, they could see individual sarcomeres, the microscopic units of muscle fiber, contracting and relaxing. They achieved a resolution of mere micrometers. They weren't just seeing shadows. They were seeing structure. Crucially, they had an undo button. When they were done imaging, they simply rinsed the mouse with water. The tartrazine diffused out of the tissue. The refractive index of the cellular water dropped back to normal. The scattering returned, and the skin became opaque again. The dye was excreted through the urine within 48 hours, and the mice showed minimal inflammation. It was a temporary, reversible window into the body. However, science is rarely a straight line to success. Following the publication in the journal Science, a significant debate erupted. Researchers Inagaki and Imai released a critique challenging the claim that live tissue was truly being made transparent in the way described. Their argument centered on the anatomy of skin. Skin has a top layer called the stratum corneum, which consists of dead, keratin-packed cells. This layer is the primary source of light scattering. The critics argued that the tartrazine was likely only clearing this dead layer, allowing us to see through it to the organs below, rather than clearing the living tissue itself. They also pointed out the problem of osmolarity. To get the refractive index high enough, the concentration of the dye had to be extremely high. This creates a salty environment that sucks water out of cells, shrinking them. While the mouse survives, there is a legitimate question about whether the cells are functioning normally during the imaging. Are we seeing truly normal biology, Or are we seeing cells in a state of shock, paused by dehydration? The critics eventually refined their stance, acknowledging that the dye did penetrate deeper than the top layer, but the concern about cell shrinkage remains valid. This nuance is important. It means this technique might be perfect for structural imaging, like finding a vein or measuring a tumor, but perhaps less perfect for studying sensitive cellular behaviors that might change when a cell is dehydrated. Despite these caveats, the future applications are staggering. If we can refine the chemistry to be gentler, or find ways to deliver it through the thicker barrier of human skin, it could revolutionize diagnostics. Imagine the painless blood draw. We all know the frustration of a nurse poking around to find a vein. A Tartrazine-based lotion could make the skin transparent, allowing phlebotomists to see the vein directly. Consider early melanoma detection. Currently, dermatologists look at the surface of a mole. If they could apply a transparent patch, they could look into the mole, measuring its depth and structure without a biopsy. It could also enhance laser therapies. Lasers used to treat port wine stains or remove tattoos often lose energy because the light scatters as it hits the skin. By clearing the skin first, the laser could be targeted with pinpoint accuracy. The main hurdle for us humans is our skin thickness. Mouse skin is thin. Ours is thick. Simply rubbing dye on your arm might not work. We might need microneedle patches to deliver the dye deep into the dermis, or use mild electrical currents to push the molecules in. It is impossible to discuss this without touching on the cultural resonance. In H.G. Wells' The Invisible Man, the protagonist discovers a way to lower the refractive index of his body to match the air. He vanishes because he doesn't interact with light. The Stanford study does the inverse. Instead of matching the air, they match the body's internal proteins. It is a reversal of the sci-fi trope, but the result is the same: the elimination of the boundary between the observer and the interior. We are used to our skin being a private wall. The idea that a simple food dye, something we eat in chips and soda, can turn that wall into a window is both fascinating and slightly unsettling. It reminds us that our opacity is just a lucky accident of physics, a fragile scatter of light that can be undone by a yellow molecule. The Tartrazine mouse is likely just the beginning. We are now in the era of dye-based optical clearing. Scientists are already hunting for other molecules, perhaps ones that work in the infrared spectrum or are more gentle on the cells. We are standing on the precipice of a new kind of medicine, where the doctor doesn't just guess what is happening under your skin, but simply wipes away the fog and looks. Biological invisibility is no longer fiction. It is a solvable physics problem, and the solution might already be sitting in your pantry. Thanks for listening to Fun Facts Online.